Introduction. Blood groups and seroantibodies has been studied to understand and identify their structure, function, and interactions. In the 19th, the blood groups were discovered by Landsteiner, whose conclusion was based on that some people red cells glutenated when mixed with plasma from other people. During his experiment, he observed that a pattern of antigen reactions was present when he combined blood serum from different individuals. Another observation that he made was that antigens on the outside of the blood cells differ between people. In other words, if blood from what he called the A group were introduced into a host of the opposite group, the host body could trigger an immunological response and perhaps cause shock or death of the host. In 1930, Karl Steiner obtained the Nobel Prize in Physiology or Medicine, and his work leads physicians to perform blood transfusion safely. He initially recognized three different blood types, A, B, and C. After many years of study, the C blood type was recognized and called type O. In 1910, the fourth blood type, AB, was discovered by one of Landsteiner's students. Around the mid-1940s, medicine developed new techniques to help us understand and identify the blood groups, and in consequence, the antiglobulin test was created and developed. As we can see in this graph, this is the direct antiglobulin test, which checks for antibodies that are attached to the surface of the red blood cells. In this test, the sensibilization of red blood cells with incomplete antibodies takes place in vivo. The cell-bound antibodies can be detected by this test, in which antiserum against human immunoglobulin is used to agglutinate patients' red cells. This new test allows the detection of no agglutinating antibodies, and this helps biochemical genetic investigations establish that oligosaccharide structures plays an essential role in determining the antigen's specificity. Blood types and correlation to diseases. To explain this part of the topic, we must talk about blood type antigens first, and we will see why. Blood antigens. The importance of the discovery of ABO antigen was huge because blood transfusion has been used since then, safely and frequently. Blood group antigens are prime important in transfusion medicine because they are the most immunogenic antigens blood group. Through our evolution, blood type is seems to be necessary because the frequencies of different ABO blood types vary among diverse population, and this can be used to suggest that a particular blood type advises a selection appendage. However, the physiological function of ABO blood type antigens are still a mystery. Okay, so let's review a little bit about blood antigens and to give a general concept about what exactly is an antigen. An antigen is any specific target to which the immune system can react. The two major antigens family important for blood types are ABO antigens and Rh factor. Let's talk about ABO antigens first, and then we will talk about Rh factor. The ABO system is comprised of three alleles for blood type. In this particular class of erythrocyte cell surface protein, the A and V alleles are codominants, and they are represented with a capital I and an exponent A, and capital I and exponent V. They are codominants alleles, and which means that a person may express one, both, or none of the ABO antigens. If the A allele over here is presented on one chromosome and the V allele is presented in the other chromosome, this person is considered to be AB blood type. The O allele is recessive to both A and V alleles and is represented with a lowercase I, which is here. People with type O do not express either variant of these proteins and have a homozygous recessive genotype. The naming system for blood types is based on present or absent of these proteins variants. The four blood types are A, B, AB, and O. Let's talk about the antigens that are present in each blood type. So we see that blood type A has A antigens and produce V antibody. Blood type B has V antigen and produce A antibodies. Blood type AB has both alleles and has A and V antigens and produce none of the antibodies. Type O has no antigens and produce A and V antigens. Rh factor. A decade after receiving his Nobel Prize, Landsteiner and his student A.S. Weiner discovered a secondary protein that also has a major impact on blood type. Their work with rhesus monkeys, we actually can see the rhesus monkeys on the right hand side of the slide. This leads to the creation of rhesus system, which highlights the presence or absence of the Rh factor. If your red blood cells has this protein, your plasma naturally contains Rh antigens, and this means that you are positive or Rh positive. If you don't have this protein, your blood is considered Rh negative or negative. Standard blood typing combines ABO system with a rhesus system to create a more complete picture of your blood profile. That means that you could have one of these A blood types. Just as an important fact, O negative it's considered to be a universal donor, means that O negative can donate blood to any of this type. AB positive, they can receive any blood type, including O negative, of course.